Okay, so I'll summarize the procedure happening in the PCR once again. PCR is responsible for carrying out the amplification of the gene which is of interest. So the gene which is of interest is first selected, but along with the gene, that particular sequence of nucleotides, you should have certain nucleotides flanking both the sides of the gene of interest. That's the only condition for carrying out PCR, right? So such a gene is selected, double-stranded DNA will be selected. There are three steps involved in the PCR. Step 1 is the denaturation, step 2 is annealing, and step 3 is again the amplification. So the three steps are happening in three different temperatures. The first step, that is the denaturation, that is you're cleaving the double-stranded DNA into two equal strands. So the two strands will be spliced off with the help of high temperature. We are elevating the temperature to about 95 degrees Celsius, and it is carried out only for a fraction of a second, that is fraction of minutes, that is 15 seconds. Only for 15 seconds this elevated temperature is carrying out the splitting up of the DNA, double-stranded DNA, so that two single-stranded DNAs, two single strands are formed. These two single strands are now acted upon by primers. They will be exposed to numerous number of primers. So the primers which are having complementary sequences will be coming and getting attached. And this annealing, this phenomenon is known as annealing. Annealing means joins. So these annealing process is happening at a reduced temperature, that is at 55 degrees Celsius. That is exactly 55 degrees Celsius, but the temperature can range between 50 to 60 degrees Celsius. So this temperature, at this temperature range, what happens? The primers comes and joins on the 3' end of the strand, so that a new 5' to 3' end sequence start getting added, right? So the new primers will be, which are so A, suppose this is A, the primer should be having GAT. If suppose GAT is the sequence, ATC will be the complementary sequence. So that should be the primer. So primer will be getting annealed at this particular temperature, and this annealing is carried out only for again a half a minute, 30 seconds. So within that time interval, what happens? Annealing happens. Specific complementary sequences comes and gets attached onto these single strands. Right now we are subjecting it for the step 3, that is polymerization. That is the more and more, that is the strand will be getting elongated. The elongation of the strand happens so that a complete DNA strand, new DNA strand, which is exactly similar to that of this parent strand, will be formed. So this is the strand, newly synthesized two daughter strands. Okay, so this is a parent strand, this is a daughter strand. So at the end, and this is happening again at an elevated temperature of around 72 degrees Celsius. So you know this temperature is always fluctuating. So these temperature fluctuations carried out by an instrument known as thermal cyclers, which is already installed in the PCR. Right. So with the help of thermal cyclers, the temperatures will be fluctuated according to the steps. Right. So first we are having an extreme temperature of 95 degrees Celsius, followed by a reduced temperature of 55 degrees Celsius. Again, the temperature is getting elevated to 72 degrees Celsius, and this polymerization is happening for about 1.5 minutes. Right. So that much is the time required for a complete cycle. So at the end of the first cycle, you will be having exactly doubled number of DNAs. That is, first we were having only a single DNA or a single gene. So this gene is undergoing duplication, or two strands are formed. Two sets of genes are formed at the end of one cycle, and each of these DNAs are again entering into the next round, so that four DNA strands will be formed, produced at the end of the second cycle. In the third cycle, we'll be having 8. In the fourth cycle, we'll be having 16. Right, so the number is getting elevated or getting multiplied in a geometric fashion. Okay, so that is how. So at above 10 cycles, at the end of the 10th cycle, we'll be having around thousand number of DNA strands. Or a single gene in the, we will be having 1000 number of daughter DNAs at the end of the 10th cycle. For 30 cycles of geometry, you'll be having billions of cells, billions of genes. So that's the advantage of PCR. So you can carry out a vast number of studies. Whatever study you have to carry out, you can carry out with a single gene by using PCR. That's the advantage of PCR. Now, there are other other applications also for PCR. So PCR is usually carried out in diagnosis of various infectious diseases like TB, then retroviral infections. That is typical examples are COVID. Then forensic medicine. Another important application is forensic medicine. Then you will be having inherited disorders like cystic fibrosis. Cystic fibrosis is a common inherited disorder which is happening in the Caucasian populations. So that like Huntington's disease. So all these diseases, inherited disorders, then thalassemia, then sickle cell anemia, etc. So all these studies, all of these are inherited disorders. That is, all are happening at the genetic level. So the genetic level studies can be carried out by using a small volume of sample is required only. But with this small volume of sample, you can amplify it and carry out the studies, right? Then prenatal diagnosis, then cancer, carcinoma detection, then transplantation, etc. Then the main application, commonly we are using it in the diagnosis of various viral infections and forensic medicine. Paternity testing, paternity dispute is there, you will be making use of the forensic PCR for carrying out the paternity. You can settle the paternity dispute exactly with the help of this PCR technique, because because we are going on looking for the gene which is responsible, right? So the paternity dispute can be settled. Then I've told you that the before PCR, the early technique we call it as a RFLP technique, restriction fragment length polymorphism, but it is now replaced with PCR. It was having certain limitations, like it was a slow process. Again, it required large quantities of DNA. Now, in case of PCR, we require only a single gene, right? So that's the main advantage of PCR over other techniques. Then this is after PCR, after a gene has been amplified, this is a restricted, we are identifying the DNA is analysis. DNA analysis can be carried out by a technique known as Southern blot technique. That is, the DNA, genomic DNA is first digested, it is denatured, it is then subjected for electrophoresis, because DNA, these are biomolecules having charged species. So based on the charge or based on the size, based on the charge, we can separate the DNA strands. So that's how we can isolate or we can identify the DNA. Then we make use of the blotting techniques. That is, simply we are blotting it in nitrocellulose paper. So it's a time consuming process, but we can identify the gene which is of interest. Then what are I've already that is they are made use of, mainly the DNA technology is made use of in the diagnosis of human diseases like Huntington disease, cystic fibrosis, Fragile X syndrome. Then gene therapy. Gene therapy means, again, we are using therapeutic cloning is carried out, right? So if there is any mutation in the gene, this gene can be manipulated by using a therapy known as human gene therapy. But there are two types of gene therapies: somatic cell therapy, somatic gene therapy, and germline therapy. Germline therapy, we are not going for germline therapy because of some ethical issues. We usually go for somatic cell therapy. And the first gene therapy was carried out in human in the year 1990. Because she was a girl, she was a 4-year girl having a SCID, severe combined immunodeficiency syndrome, known as adenosine deaminase deficiency, ADA deficiency. So this ADA is very much responsible for carrying out the DNA synthesis. DNA for the synthesis of purines, purine metabolism, purine synthesis. One of the most important enzyme which is responsible for carrying out the purine biosynthesis by de novo pathway is the adenosine deaminase deficiency. So the girl was born with ADA problem. ADA deficiency was there, resulting in severe combined immunodeficiency disorders. So this was corrected by making use of gene therapy. And that girl, I've got the photo of that girl. This is Ashanti De Silva. She was treated. She's completely cured. So this is one of the most important landmark in the history of recombinant DNA technology. That is, the first FDA approved gene therapy was carried out in the US for ADA deficiency. So again, we are having two strategies. Somatic cell therapy can be carried out, and germline, but this germline therapy is not usually carried out because it's mainly carried out on experimental animals. And there again, the same, the recombinant DNA is synthesized, recombinant DNA is made and is introduced into vectors. The vectors we usually use are retroviruses or adenoviruses. Again, retroviruses may be replaced with adenoviruses because they cannot produce, adenovirus do not produce any mutations. They cannot induce any mutations, but retroviruses are having a tendency to create mutation in the host cell. So sometimes the retrovirus may be replaced with adenovirus. Right, so we are the recombinant DNA, that is the gene of interest is taken, it is introduced into a circular DNA, and this recombinant DNA we are now introducing into a viral vector. So this virus can be retrovirus or it can be adenovirus, or other organisms can also be used, but the commonly used ones are the retrovirus or the adenoviruses. And this again, among the retro and the adenoviruses, the most important advantage of adenovirus is that they do not cause any harmful mutations during the integration, but it's only a transient phenomenon. But in case of retrovirus, once the gene is introduced through the retrovirus, it will be transmitted to the progeny of the cells also. That is, throughout the life, it will be sustained. But in case of the adenovirus, the expression will be only for a short period of time. Again, there are certain strategies for carrying out the gene therapy, because it's not that easy for carrying out gene therapy in humans also, because it is under the strict guidelines of National Institute of Health. And for a person to be subjected for gene therapy, there are certain conditions to be fulfilled. That is, the gene should be able to be cloned and should be well characterized, and there should be some effective method for the delivery of the gene into the tissue or to the humans. And again, the most important factor is that the risk posed by the gene therapy must be least, right? It should be minimal in nature, and you will be going for a gene therapy only if it is proved that the disease must be not treatable by any other mode, any other strategies. Then only you can go for gene therapy, right? And another important, so we have gone for PCR, DNA, nucleic acid based technology. We can make use of amplification of the DNA, and you can manipulate the gene. You can make the gene corrected and can be introduced into the person, and we can treat the inherited disorders. So that's one of the most important application of the DNA based technology. Another important application is that, can you identify this picture? What is this demolished building? You have got any idea? Where did it happen? When did it happen? It was one of the most important events happened in the US approximately 20 years back. The 9/11 attack, ground zero, right? So this is the much more clear picture. That was a, yes, Pentagon, and this is the ground zero, right? So what is the relation between this RDNA technology and ground zero, 9/11 attack? Around numerous number of people has lost their life in this incident, right? And it was able to identify those persons who have lost their lives with the help of our DNA technology, and the technology, that process, that phenomenon, the technique, we call it as DNA profiling. And this DNA profiling was first again developed by a scientist named Sir Alec Jeffreys in the year 1984. And what is DNA profiling? Maybe DNA profiling, rather than DNA profiling, we will be much familiar with another term which is known as DNA fingerprinting, right? DNA fingerprinting, that's a much common name for that, but DNA fingerprinting, that name is now replaced with DNA profiling. So what is DNA profiling? It is using a DNA sequence data for the identity, for identifying a person. So that is what you call as DNA fingerprinting. You know that the fingerprints of no two persons will be similar to each other, except in the case of identical twins. The same way, the DNA of two persons cannot be identical in nature. Of course, the genome sequences of all the members of a same species are almost similar, but there are certain polymorphisms happening which make one individual differ from that of the other individuals, right? So that polymorphism, that is we are making use of.